Genes. While we all know that they are what determine our traits and how unique we are to one another, do you know of the complex process that allows for that to happen? This research all started on a pea farm in what is now the Czech Republic in the year 1856 with a man named Gregor Mendel. Good evening, Mr. Mendel. It is so lovely to meet you.
You as well. I seriously cannot thank you enough for having me on your show today.
Now, tell me, how does it feel to be in a time when your research has become such a foundation for genetic studies all over the globe?
Well, I must admit, it was rather shocking at first. I had always hoped that my research would become something greater than I could ever imagine, and that's definitely what's happened. It has felt so rewarding to learn about things in my field only possible with modern technology.
Oh, I'm sure. When you say your field, what are you referring to?
By that, I mean the study of genetics, which is what you now call it. I've spent years of my life slowly studying how traits are passed down from parent to offspring, and can only discover a small percentage of what you know today.
Your discoveries are one of the main reasons we are so advanced in genetic research. Would you mind giving further details on some of the concepts you discovered?
Not at all. So, first, each parent gives one factor to the offspring. Those factors then determine their traits. These factors will pair up, sometimes being dominant over another one and masking each other. I call that the law of segregation. The law of independent assortment says that the factors of different traits do not affect one another.
That is truly fascinating. How long did you say you were working on that for?
It was 8 years.
Wow, that, ladies and gentlemen, that is dedication.
Guys,
Well, if you don't mind, I'd love to educate you on everything we've learned in relation to your research since the 1800s.
Oh, please.
Perfect. So, first, I'd like to start with cells. All living things on Earth are made up of these tiny units called cells. Inside of these cells, hereditary information is stored. DNA is a molecule that carries hereditary information.
Hereditary as in information that will be passed down?
Exactly that. The factors that you discovered are actually a small segment of DNA called genes.
So that means genes are what determine and control traits?
Correct. Genes can also have different forms called alleles. They allow for more variation in the gene and dominant and recessive traits, as you described. That brings us to phenotypes and genotypes. Phenotypes are all of the observable traits. Some examples are whether you're tall or short, whether you have brown or blonde hair, or whether you have freckles or no freckles. Genotypes refer to the genetic makeup of an organism. Each letter in the genotype is an allele. An uppercase letter means that allele is dominant, while a lowercase letter means that allele is recessive. When both letters are in the same case, they are homozygous, so they can have homozygous dominant and homozygous recessive. When there is one uppercase and one lowercase, it is heterozygous, though the dominant allele will dominate the recessive.
That's cool and all, but when are genotypes used?
Genotypes are used in a Punnett square. They are used to determine the possible combination of alleles in an offspring by using the parent's alleles.
So, it seems like I got most of it.
Not quite. DNA is then condensed into thread-like structures located in the cells called chromosomes. This means that genes sit on them too. Each organism has a certain amount of chromosome pairs called homologous chromosomes. Us humans have 23 homologous pairs.
That is a lot to take in.
It can be, but it gets less complicated when you fully understand the process. To make cells for reproduction, the organism's cells must undergo meiosis. We'll come back to that later. During meiosis, the chromosome is able to swap pieces and create new genetic combinations.
This is truly riveting. How could I have missed so much?
Since cells are microscopic, it is impossible to see them without modern tools. That is likely why you thought all traits follow independent assortment, which is incorrect because some traits tend to be inherited together. Some also have multiple alleles, making dominant recessive patterns a little bit more complicated. Some dominant and recessive alleles can blend, and other times, two dominant alleles can both be equally present.
Everything seems more complicated than I had initially thought.
That may be true, but it just shows how precise our bodies have to be when passing down traits. Giving one too many or one too little chromosomes during meiosis can lead to disorders, or even death.
So, about that meiosis thing, what is it?
Meiosis is the process that makes reproductive cells. In humans, it makes sperm and egg cells called gametes. There are two phases of meiosis. Meiosis 1 is called the reduction division. The cell starts with a diploid number of 4, meaning there are 4 homologous chromosomes in the cell. It all starts with interphase, when all of the genetic material in the cell has just been duplicated. In prophase 1, the DNA is finally condensed into homologous chromosomes. In metaphase 1, all the homologous chromosomes line up at the equator of the cell. This is where crossing over can happen. In anaphase 1, the homologous chromosomes are pulled to opposite sides, while the sister chromatids stay together. In telophase 1, the cell begins to split into 2, and by cytokinesis, each new daughter cell has half the chromosomes the original cell had. Now we're on to meiosis 2, which both daughter cells will go through. Prophase 2 is more or less the same as prophase 1, just with half the amount of chromosomes. In metaphase 2, the chromosomes will again line up at the equator of the cell, this time splitting. After the sister chromatids split, each chromatid will be pulled to either side of the cell. In telophase 2, the cell is yet again being split. At the end of meiosis, you'll be left with 4 haploid egg or sperm cells, each ready to meet with other haploid egg or sperm cells.
I guess I was pretty far off from the truth.
Not really. Your research is the only reason we know any of this. Without your findings in the inheritance of traits, who knows how much less advanced society would be in the genetics field.
You're right. I'm proud of my research and how far it got you.
You should be. Now, I think it's time for a little game.
A game?
I will recite a statement that you have previously made about genetics. On the whiteboard provided, write down if that statement is correct or incorrect. Do you understand?
I understand. Ready whenever you are.
Perfect. The first statement is, traits are passed down in units. And your answer is? Correct. Wonderfully done. You may now erase your board. The second statement is, traits are all sorted independently. And your answer is? Incorrect. That's right. Your third and final statement is, there is one trait inherited from each parent. And your final answer is? Correct! You got it, Gregor. On that note, we will be back to you with a career specialist after this short break. I am a career specialist.
Seriously? I had no idea.
Now, from my professional perspective, I believe that the career meant for you is a...
What is a genomics researcher?
I believe that you will love it, Mr. Mendel. A genomics researcher is someone who studies the effects of genes on physical traits and how certain combinations of genes influence disease, growth and development of organisms, and the response to the environment.
That does sound
interesting.
I knew you'd say that. Just think, you get to be making big discoveries and doing experiments to get there all the time in this profession.
That would be a dream for me.
On top of finding you a possible career path, Gregor, we are also going to share an area of genetic biotechnology with you.
Hold on, what even is that?
Oh, right. Genetics biotechnology involves manipulating genetic material, DNA, to introduce, remove, or modify specific traits.
That is insane. Though, it does sound like there would be a lot of hurdles in its path.
That's for sure. One example of genetic biotechnology is genetically engineered crops that have been modified to resist pests, tolerate herbicides, or to increase nutritional value.
No way!
Yup, it actually first happened over 30 years ago in the mid-1990s. It allows for better nutrition and more reliable food sources. Not only that, but genetically modified crops can help reduce hunger in poorer regions while reducing pesticide use at the same time.
That sounds perfect.
Actually, it does bring on some issues.
I suppose you can't have everything.
For starters, not everywhere can afford the technology, which would make poorer places even more poor in agricultural terms. Some people also do not like the idea of eating genetically modified food. Other people also don't know when the food they're eating has been modified. On top of that, the lack of pests could harm beneficial insects and spread to wild plants, both affecting the ecosystem very negatively.
You're right. Thank you for teaching me about all of this.
It was no problem. Thank you for coming. I think that on that note, we can close off the show. That was Gregor Mendel, everybody. Have an amazing evening.